Hi, year 11s and 12s. This is just a short video talking about how to do a deconstruct and design practical. So I've got the task sheet up here and we'll look at 3 different exemplars. I'm just going to go through part A for this task, which is to research, deconstruct the problem and design your own experiment. I'm not going to go through part B, which is to complete the experiment. You know how to complete the experiment, plus it's going to be the experiment you've designed. And I'm not going to go through part C, which is write an individual report, because I made a video on that one for your first practical, which was how to write up a practical investigation, and we looked at the exemplar as well as had on the side the task sheet and how to answer that accordingly. So first off, let's talk a little bit about what this experiment actually is. So it's based on topic 3, which is homeostasis. It does touch a little bit on topic 2, cells as well, because we're talking about what makes an organism respire faster. Now this is your overall question. What makes an organism respire faster? Okay, so we know that all living things go through some sort of respiration, whether it's aerobic or anaerobic respiration. Okay, and those reactions help to generate ATP and help the organism gain energy. Now, there's lots of different ways that we can measure cellular respiration. So we could do volume, or the amount of oxygen taken in, volume, amount of carbon dioxide being released, lots of different things that you'll need to research. Okay, we've got some equipment at the school, which we'll go through in lessons showing you the types of equipment that we have, whether it be aerobic or anaerobic, that we can measure respiration. But this will be something you need to design. Okay. So your task in this entire practical, to design an investigation to test the effect of one factor on the rate of respiration in an organism. To investigate the question, what makes an organism respire faster? You're going to need to do research, determine what types of organisms you could use, think sensibly. There's only going to be a certain amount of organisms we can purchase at the school that you can use. Think about ethics, safety. Is it going to be easy to work with that organism? How are you going to measure that organism's respiration to test your hypothesis? You're going to need to test a whole lot of different things. Okay, so you're going to need to think of what organism am I going to use. Am I going to use an animal? Am I going to use a plant? Am I going to use a seed? What factor is going to affect respiration? Okay, am I going to look at things like temperature? Am I going to look at any sort of things you could, pH maybe? I don't know if there's a link there. Maybe fear. How could you measure fear? Lots of different things that you could do to test the rate of respiration. How are you going to measure that rate of respiration? What equipment are you going to use? Are you going to look at anaerobic or aerobic respiration? Other variables. Okay. Once you've deconstructed this problem and you've thought about what you're going to physically do, that's when you need to design a practical that you would like to do in the classroom, and then with a partner you'll decide if you're going to do your prac or the partner's prac. So this needs to be an individual deconstruction, individual design. You can't design it with a partner, but when you do the experiment, you'll only do one of the experiments. So first off, you need to do some research. So using numerous sources, research the factors that affect respiration. So what are some ways you could measure respiration? What other considerations you need to look at? Keep a list of references. You'll need them for part C when you write up the physical prac. But the main thing here is we want to deconstruct the problem. So brainstorm the various aspects of the question. What makes an organism respire faster? You could do this via a table. So we've given you a sample table that you could use. Which is, so you've got all your considerations. So, for example, your overall question, what makes an organism respire faster? Then you need to think about what types of organism you could use, what types of respiration you're going to look at, what are the methods you could use, how you're going to measure that respiration, and what other variables would you need to consider. So things like controlled variables or uncontrolled variables you wouldn't be able to control. So first off, in your options, you should list all of the options for organism, all the options for respiration, all of the options for the method that you could use, all the options for measurement of respiration, all the options for controlled variables, uncontrolled variables, etc. Okay. Then you start to think of questions related to these. Okay. So, for example, for the type of organisms, what's some questions that you could research that would help you decide which organism to use? Underneath there, you go through findings. What are the findings from those questions to research once you've done some research on them, and what's some limitations of using each of those options? Okay, and you do the same thing for each of these, for type of respiration, method options, etc. You do the same thing. You fill out all these boxes here, and we'll go through an exemplar. Okay. Lastly, once you've finished that deconstruction table, now this table really should not be any longer than a page. One A4 page. I'd have it on landscape view rather than portrait, but one A4 page. Then you get to your design. and this is the reason that I only want the deconstruction table to be one page, is that your entire design practical, so what you submit, can only be a maximum of 4 sides of A4 page. Okay, so 4 pieces of A4 paper, single-sided. You could do 2 double-sided if you wish, but you can only have 4 pages total. Okay, if 1 page is your deconstruction table, the remaining 3 pages are where you're actually doing the design experiment. So where you're talking about your aim, your hypothesis, your variables, your materials and equipment that's required, your method suitable to test the hypothesis, and a blank data table. Okay, this is your experiment. You're designing it. You need to be writing this up so that this is what you and your partner will use if you undertake this experiment. Okay. We'll give you time in class to work on that, of course, and we'll help you along the lines. You'll get a draft for this, then we'll give it back to you, and once you've got the draft back, that's when we'll start to organise what we need for the experiment, and we'll send it to Mr. Harris, and we can book the experiment in. Okay, but for now, let's look at some exemplars of what we've done. So this is a first example. Now, for this person, they took the table, but they kind of turned it into their own document. Actually, before we do that, I'm just going to go very quickly. This is a very important point here. This is the most important. This is going to really set the A-pluses apart from the rest of the group. You need to annotate your deconstruction and your design experiment, so the whole 4 pages, to justify the decisions you have made. Okay, so why have you made those decisions? Why are you using that organism? Why are you using this method? You need to have justification there. What SACE is looking for, what I'm going to be looking for, is that you've got that justification there. And you'll see in these exemplars that I'm about to show you that all of those students did that. And these 3 exemplars that I'm going to show you, they're all A or A+. Okay, so this is the first exemplar. For this student, they took the table, but they put their own spin on it. Okay, so it doesn't look like the same table we gave. It's got the same things: potential organisms, type of respiration, method options, measurement of respiration, and other variables. It's got the same thing, and they've just listed the possible options there. But then they've made research questions, finding limitations, their own section down here, with questions, with findings, and they've put their own spin on that. Now, you can pause the video and have a read of this if you want to. I'm not going to read it all out to you, but no, they've got the deconstruction table there. At the bottom of there, once they've got all these options and they've highlighted, so this student wanted to work with earthworms, with aerobic respiration, they're going to look at temperature, they're going to look at carbon dioxide concentration with a carbon dioxide sensor, and the control variables they thought was good were time, size of container, size of organism. Okay. Then they've given justification based on the findings they've done here, the justifications of why those highlighted variables in the options have been chosen. Okay, so they've physically got that justification there. Once they've done that deconstruction, we've got another 3 pages on this prac where they've gone through and designed their experiment. They've got their aim, they've got their hypothesis, they've got their variables, a lot of controlled variables, how they're controlled, why it's controlled, and down here, again, we've got more justification. Okay, you've got your apparatus. Okay, why has that apparatus been chosen? They've got justification there. The method. Why has the method been chosen? Justification for the method. The student has clearly shown with annotations why they've done each thing. Safety and ethics. Still need to do that for a design experiment. Why have we gone through those safety and ethics? And lastly, you've got your blank table. So this is, you've put thought into how your result's going to look, and you've designed a table that's going to collect the data. Now, it might not be the final table you used, but it's shown us that you've thought about how you're going to collect that data. Okay, so the biggest thing for it, they're just designing an experiment, but they've gone to the depth of showing justification, and they've shown the thought reasoning behind why they've chosen these specific options. Now, the same thing with exemplar 2. Now, with exemplar 2, the student has just used the table that we've provided, but they've really filled it out with depth. Okay, same thing, they've highlighted the options they've chosen. There's a lot more research in there. Okay, then they're going for the justifications of why they've chosen each of their chosen options. So why have they chosen that organism, why have they chosen that independent variable, why have they chosen that dependent variable, etc. Okay, they've then gone through and they've designed the prac like the other student. Okay, aim, hypothesis, variables, again some more justification here in yellow. Again, materials, justification for the materials, justification for the method with a diagram. They've drawn how they expect this experiment will go, which is excellent inclusion. Obviously, space is important. Safety and ethics, and again, data table. Okay, this student got an A+, they've got their references as well. Student got an A+, as did the last student. Okay, exemplar 1 and exemplar 2 both got A+. The key thing here is the justification. As well as doing a fantastic design, the justification is there. You need to have those justifications there. Lastly, we'll look at exemplar 3. Now, this student got an A. Okay, again, they've just used the table that we've given them, and they've highlighted some possible options. There's lots of research there. Again, the key highlight in this design is justification, and they've highlighted in yellow. So controlled variables. Why is it constant? How is it constant? Why have I chosen this controlled variable? Why have I chosen this apparatus with a photoshopped version of what the diagram might look like? Why have I chosen this method? Why have I chosen the safety and ethics? Okay. The key thing is that justification. That is what you need to be showing. And even if we look at the design over here, critically deconstructs the problem and designs a logical, coherent, detailed biological investigation, you must have that justification to do well. So this first part, this IAE1, is all that's being marked in this first design prac. Everything else will be designed in the second half of the experiment, part C. Part A is just looking at design of an experiment and showing that justification. These exemplars will be put up as PDFs on the OneDrive for you to look at if you'd like to go through them.